May I know when the session will start?
Hello everyone, we shall start in a minute. I hope I am audible. Am I audible? Can anybody reply?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay, I hope my slides are visible.
Yes, ma'am. What?
Okay, so at any time, if my slides are not visible or if I am not audible, please let me know. I will be in the presentation mode. So turn on your mics and answer or tell yes or no because I won't be seeing the chat box.
Yes, ma'am. Visible everything.
Okay, thank you. Okay, so welcome to NPTEL Cell Culture Technologies. This is week 4 live session. So this will be helpful to clear your doubts or will be helpful to solve your assignment questions. I'm Sumati Raviraj from University of Hyderabad and the course instructor for this is Professor Manik Das from IIT Kanpur. So this week, the topics that have been covered by the professor is poly D lysine deposition, surface chemical analysis, cell growth process, cell surface interface, and cell culture substrate patterning. So we shall directly go into the questions this time because we can discuss a lot within the questions itself. So the questions this time are a mix of uh, few questions made by me, which are similar to your assignment questions, and few questions are direct questions from your assignment. So, any way it should be helpful for you all. Okay. So for this now you all have to answer the questions. So you can turn on your mic and say the options, whatever you think is the answer. So the first question is, what is poly D lysine used as in cell culture? A. Protein source. B. Nitrogen source. C. Adhesion on surface. D. Suspending the cells. Anybody who wants to answer? Anyone? So this is supposed to be an interactive session. So you can let me know whether you know about it, whether you don't know. What do you think could be the answer? Because this is already discussed in the
Nitrogen
previous session.
source.
Okay, somebody said nitrogen source. Anyone else? No. Okay. So nitrogen source is not the correct answer, but good for at least trying to guess. So the answer is adhesion on the surface. Like we discussed previously in the previous interactive sessions or in the videos, you might have seen that when we culture the cells on a petri dish, so sometimes we have adherent cells and sometimes we have non-adherent cells. So these cells, this is your petri dish and this is your cells. Okay, so these are your cells. So the cells need a surface for attachment. That is when they are adherent cells. So these types of cells, if they are highly adherent, they will go and attach to the surface. And if they are semi-adherent, like we discussed, there are adherent, there are semi-adherent, there are completely suspension cells which do not adhere at all. So semi-adherent means cells which have the capacity to adhere, but due to any light stress or any disturbances or while changing the media, they can detach from your plates. So for such kind of cells, there is a definite need for a surfaced adhesion molecule, because your epithelial cells, your semi-adherent and your adherent cells are epithelial like cells or endothelial like cells which need a base to adhere. So when they want to adhere, they need some molecules on which they can go and sit. Only then their attachment will be strong in that. So for these type of cells, we need to use poly D lysine coating. These are synthetic coatings. So poly D lysine will have be coated on your petri dish or your glass cover slips if you're growing cells on the glass cover slips. So they will, this is charged base attraction. So your lysine will have positive charge and your cells will have negative charge on the membrane. So those positive and negative will attract each other and it will help in holding the cells on the surface. Okay. So mostly whatever plates we use, cell culture dishes we use in the lab for culturing the cells will be TC treated, that is tissue culture treated plates. So these plates will help in adherence of your cells. So polylysine. Why polylysine? Because as I said, polylysine is a synthetic positively charged polymer existing as two enantiomers. So one is poly D lysine and one is poly L lysine. Both are commonly used. However, poly D lysine is not degraded by cellular proteases and is therefore often preferred choice. Okay. So you can use poly L lysine also. Many people use poly L lysine, but since lysine, poly D lysine or poly L lysine is what? Nothing but a polymer of lysine. Lysine is an amino acid. So many amino acids attached together form this polymeric chain of this poly D lysine or poly L lysine. So because they are amino acids, they are not foreign to your cells, right? So your few type of cells may have some kind of proteases which might degrade this lysine. So but these will degrade only preferentially L type of lysine, and most cases D type of lysine will not be degraded. So that's why poly D lysine is better preferred in case of cells which might have this capacity to cleave the lysine residues. That is your L lysine. So your L lysine and D lysine are nothing but enantiomers. So they are the same molecules, they are the same amino acids, but they are like mirror images. They are like your left hand and your right hand. Okay. So they are the same thing. They have the same structure. So this is your structure of your lysine. This is your lysine, and this is your polylysine. Okay. So this part within the bracket is your lysine. So if many lysines like this attach to each other, which is denoted as N means N number, many number. So then it will become your polylysine. So this is, see, you can see here it's depicted like this lines. So if you put a mirror here, if you have a mirror image of this, then your L lysine will be D lysine or D lysine will L lysine. So this is how you can understand how they are. But what actually they do is, see, so what is your enantiomeric property of the molecule is its ability to rotate the light. So your light, your normal light, your light source will have uh, play will be in different planes. Okay, so your light, you should be knowing that it will have both electric field and magnetic field. It's a electromagnetic field. So the arrows that you're seeing here, so those are depicting your electrical fields. So your electrical fields will be going, like, you know, the nature of light, right? It's both uh, particle, and it has wave nature as well. So what you're depicting as your wave nature is nothing but your electrical capacity of the light. Okay, so these will be usually, if it's your normal light, it will be in all the directions. So your electric field will go in all the directions. But when you put a polarizer is nothing but which passes, which filters your light, and it will allow only light in a one direction or one plane. So that is called polarize. So you can know with the name itself that it will polarize your lights. Polarizing means one direction, one plane. So when you place it under a plane, then you will get a single plane light which has single plane. So then when you put some molecules in this is a chemical property. So when you put some molecules in this, where your plane polarized light is entering, your molecule has the ability to rotate the light. So when your plane polarized light is hitting your molecule will rotate your light. It will rotate either like this or like this. So if it is clockwise, it is known as dextrorotatory. It is anti-clockwise, it is known as levorotatory. So that is only your D and L. So dextro is D, levo is L. So based on this property of your molecule to rotate the plane polarized light to either your clockwise direction or anti-clockwise direction, they are classified as D lysine and L lysine. So it's not just that they are same molecules, they just are like mirror images. They have the specific property, and because of this property, they can also have specific different functions, or they can have specific stability. Like here you can see L lysine is not as stable when acted upon by proteases like how D lysine is. So D lysine has more stability because it is dextrorotatory. Okay, and so this is how you will get to know whether it is, you know, dextro or levo rotatory. So what else you can know here is few cells like your neuronal cells or your cortical neurons, there has to be a particular concentration of this poly D lysine. It's not like you can use whatever quantity you want. Like everything that you use in a laboratory, it has to be optimized and it has to be given in a certain standard range or standard quantity. So why that is, you can see here, this is one of the papers. So you can, if you want to know more about it, you can visit this. So yeah, so what happens here is, this is your concentration of your poly D lysine. So this bar shows this is highest concentration and this region is your lowest concentration. So when you start increasing, this is increasing concentration. So your 1, 2, 3, 4, you can see the color gradient. This is least concentration of D lysine. This is highest concentration of poly D lysine. So as your poly D lysine concentration increases, what you would expect, like if you didn't know, you would think, oh, it helps in adhesion of the cells. So if I increase more poly D lysine concentration, more the cells will attach. So you will think it's like a proportional concept, that more lysine, more adhesion. It's not like that. So there is a particular optimal range only where it will adhere properly. So here you can see at the least quantity the cells are not adherent, but at the same time in the highest quantity the cells, you can see they are not properly adherent. Even though they might go and stick to the plate, they are not properly adherent, which means they will not uh, spread out and show their specific morphology. Like how you can see in 2 and 3, you can see that these circular cells, whenever they're in suspension, cells will always be circular, spherical, regardless of their actual morphology. So you can see when there is no D lysine, the cells are in clumps and they are in spherical form, but when you increase your D-lysine concentration, they go and they attain the neuronal shape, the neuronal morphology in 2 and 3. But when you increase it more than 3, that time again they get spherical. That means their attachment is improper. So this is your optimum concentration. Okay. And also for cells like neurons or some other differentiating cells, where the cell has to attach, spread out to its normal morphology, and then differentiate to produce its own proteins or start gene transcription, translation, and then become a differentiated cell. So there this is more important, because if there is improper concentration of poly D lysine, your cell will not mature or differentiate. Both are similar. So you can see in the least concentration also there is no maturation. In the highest concentration also there is no maturation, because in the highest concentration, they are, even though they are settled down, they are not properly adherent. Okay. So in your 2 and 3 only you can see proper maturation. In your 3 you can see proper maturation. In your 2 uh, there is still probability to mature, but it's not complete. They are attached, they have shown their complete morphology, but maturation is incomplete. So this is like a combination. In 1, no attachment, no maturation. In 2, attachment but no maturation. In 3, attachment as well as maturation. That is what we want. So 3 is your correct concentration of your poly D lysine, whereas in 4, improper attachment and no maturation. Okay, I hope it was understandable. If you have any doubt, you can ask me at any time. Okay, so shall we go to the next question? So the next question is, how do we improve cell adhesion, proliferation, and growth of cells in culture dishes to better mimic the physiological state? Which means when you add cells to grow, as I said, sometimes they don't adhere, sometimes they don't proliferate. I told you here, they, even though they adhere, they don't mature. So if the adherence also should be properly done, the maturation also should be properly done. For that, it has to grow properly, produce correct proteins, and then it will only attain a proper maturation when when it is most similar to how it was in your body. So that is nothing but mimicking your physiological body state. So when will the cells be able to do all this? Like, how do you improve this condition? How do you improve cell adhesion, proliferation, growth of cells? How can you help yourself to become maximum similar or identical to the cells when they were in your body? So what can you use to improve this condition? First option is increasing growth medium. Second is increasing glucose. Third is ECM coating. Third is change in temperature. What could be the answer? Anybody? Come on, sir. Somebody tell me.
Uh, HCM cutting.
Yes, very good. So the answer for this is ECM coating. So just like how we saw in the previous question, poly D lysine is a coating. Okay, it is a synthetic coating. So in the same way, if you have this, it will help yourself to adhere more, proliferate more, grow better, and it will be more similar to how it was in the physiological condition. So answer is ECM coating. So what exactly is ECM? ECM is nothing but extracellular matrix, which is anything that your cell needs, or which is surrounded by your cell. And when you even plate or seed the cells in your cell culture dishes, after a few hours or after a day or two, they will start secreting extracellular matrix by themselves also, so that they can start complete attachment, forming their proper shape and to differentiate. So in your body also, you have this extracellular matrix which will help your cell attach properly, and also have connections with different types of cells wherever it needs, and then get some signals also. Many of your proteins are secreted in your extracellular matrix, and via your extracellular matrix, the other cells receive this protein. So all your signaling also happens like this. So here you can see these are your epithelial cells. So you can see here your tiny dots on your epithelial cells. These are integrins. So these are the molecules which are present on your cells, which help in adherence of your cell to the extracellular matrix or any other protein in that matrix. So your extracellular matrix will have many different proteins depending on the cell type, depending on the tissue type, and depending on the animal also. So it will have major proteins like proteoglycans, collagen or laminin, and then you see there is a connective tissue also here. So then around the connective tissue there is fibronectin, fibroblast. So all these will be present in your extracellular matrix. So mostly the proteoglycan, collagen, laminin you will see on your basement membrane where your cell needs to attach. So your cell will have your integrins, your basement membrane will have your proteoglycans and collagen and laminin. So integrins will attach to these. Okay. With the help of this attachment, your cell will stay in that particular position. Okay. So this is how it is in your body. Now in your in vitro conditions, you cannot have all these things. Like you can have, there is also a cocktail or mixture of these kind of matrices. But usually you do not require all of the things for complete adherence. It depends on what type of experiment you're doing, what type of cell it is. Based on that also you can choose your coating on the Petri dish. So why this is very important is you can see here in your classical 2D environment, that is nothing but your cell culture plate or your 96 well plate or your 100 dish or T25 flask. You have your bottom, you have your plastic, usually it's a plastic or a glass bottom. So that time it is when it is not coated with anything, your cell, if it is a very adherent cell, even without all this it will go and attach. But when I was talking to you about improper morphology, it will not spread properly and all. So that is what is depicted here. Here you can see it has just gone and it is lying flat on the surface. Okay, and then the cell body is on top. It's not attaining any shape. It's not, spreading out. It's not doing anything. But when you have a physiological 2D environment where you have some biological or a chemical coating, so that time what happens is, see, you can see your cell has attained some morphology. It has some structures, some protrusions coming out of it. Usually adherent cells should have these kind of extensions coming out of your cell body, because those extension help in your cell to hold on and attach properly on the plate. And sometimes it will also help in migration of the cells if there is some specific conditions where you want to see the migration capacity of the cell, those kind of things. So this kind of coating will help your cells attain a better physiological state. So the some examples of this coating that you can do are like here ECM proteins. Like I said, laminin you can use, fibronectin you can use. There are collagen coated plates, gelatin, vitronectin, etc. And then there are chemical and synthetic coating, like we discussed before, poly L lysine or poly D lysine, and also polyornithine. This is also amino acid. So these kind of poly uh, lysine and ornithines are your chemical and synthetic coatings. Next question. Which can mimic the collagenous environment of cell attachment in vitro? So, collagenous environment. So, what the question means, what is similar to your collagenous environment for attachment of cell? Which one is similar to collagen basically? So A glucose, B laminin, C gelatin, D lysine. Anyone?
Gelatin.
Gelatin. Very good. So your gelatin is nothing but a derivative of your collagen. Okay. So that is why it is very similar to collagen. So how you can use gelatin is, you can either have a solution of gelatin, you can place like the required volume based on the surface area of your plate. You can add that volume in your plate, you can spread it around properly in sterile environment, and then you can leave it at room temperature for some 20 minutes, 15 to 20 minutes, and then you can remove all your excess gelatin. Then you will have a nice coating of gelatin on your plate. So all this should be done in your sterile environment. Then based on your manufacturer's instruction, you can either wash it with PBS or do whatever steps that is required. Other than this, there are this newly ready to use gelatin uh, patches that are available. So these are non-woven, and on these kind of non-woven thread-like structures, they already put gelatin and then they freeze dry it. So when they freeze dry it, it maintains its structure, and then after that they will supply it to you, and then you can take this sheet and you can put it on your, see, this is how the sheet looks like when you take it out. So you can take it and place it on your cell culture dish. No need to do anything else, and then it will attach to your plate. So once you add media, it will all get settled down, and then you can grow your cells on this. So if you look at microscopical view, this is how it will look. Tiny, tiny thread-like weaves on which there is a layer of gelatin. So your cells, here you can see your blue color stained are your cells, and these are your gelatin woven sheets. So your cells will attach to this gelatin and then they will grow happily. So gelatin is a hydrocolloid and is rich in glycine, proline and hydroxyproline, which imparts structural stability. So gelatin is a hydrocolloid, means it's a hydrolyzed form of your collagen. Okay. So gelatin is also used in other applications. So other than just coating on the plates where you can use it as a basement for attaching your cells, there are other applications for which you can use it. You can use it to stabilize the tag DNA in PCR. So in PCR you use tag DNA. So for that you can use gelatin to stabilize it. And blocking reagent in Western blotting, ELISA and immunohistochemistry. So in Western blotting, ELISA and immunohistochemistry, there is generally a step of blocking. Blocking is done to prevent unnecessary or unwanted attachment of your secondary antibody on directly on your either NC membrane or your ELISA plate or your slide with your tissue sections. So we usually do it either with normal goat serum, that is one thing, or you can do it with skimmed milk. So usually we go with skimmed milk, but you can also use gelatin. So this will cover all the areas where your secondary antibody has a potential binding site, maybe. So that time what happens, it will decrease your background, unnecessary or unwanted bands. So your secondary antibody is supposed to bind to your primary only. So if it does not, if it also binds to some other area on your blots or on your ELISA plate other than where your primary antibody is, then you will get some false results or some extra false bands which are unnecessary. So for that you can use this to prevent that. Then it can also be used as a component of media for species differentiation in bacteriology. So in bacteriology, when you grow bacterial cells, so that time you also use gelatin in your media. So it will help in your growth, better growth of your bacterial cells. Then it is a biocompatible polymer, and then it is a delivery vehicle for active biomolecules. So since it is a biocompatible polymer, it is derived from collagen itself. So you can use this as a delivery system for for some drugs or some biomolecules, very small tiny molecules which you want to deliver into the ECM or anything which will later be taken up by your cells. So then it can be used as scaffolds for tissue engineering. So here you saw it for cells, like a sheet it is. So your cells will grow on the sheet like a monolayer. But tissue engineering, if you want to build a tissue from cells, or if you want to, have this tissue culture, like you want two or three more types of cells within the same, you want to have a base for all of this. So that time you can use this gelatin, and then there will also be arrangements of this gelatin. So you can have just a gel like this, or you can have like compartmentalized gelatin, where at specific compartments you will have an ordered aligned growth of cells. Okay, so these are all the uses of gelatin. So if anybody has any questions, they can ask me anytime. Okay, so how this binding takes place, how this gelatin helps in binding. So here you can see integrin mediated binding of collagen and gel. So this is your collagen how it binds, and this is your gelatin how it binds. So here your magnesium plays a very important role. It coordinates the bond formation between your either collagen gel or your gelatin to your cells. So as we said earlier, your cells will have integrins attached to them. So these integrins will bind to the gelatin or your collagen molecules in their carboxylic groups. So at this region, you will have certain sequences of amino acids which are like the binding sites. Like in the previous, maybe in your first or second assignment, you would have had a question of this RGD. What is RGD, or what RGD stands for? So if any of you remember, RGD is arginyl, uh, arginyl glycine aspartic acid. So this is your three amino acid sequence where this binding takes place. This is your binding site. So this carboxylic groups and your Mg facilitates this binding. So your Mg plus your cations or your other divalent ions, all these help as cofactors and help in binding stability, or provide means to bond formation. So here you can see your Mg is at the center helping the bond formation, and this is your alpha subunit, and this is your collagen. So this integrin is binding to your collagen layer. Okay, so in the same way, this is your gelatin, and then this is your RGD. These are your Mg plus 2 ions helping in the bond formation. So your integrin will form bonds with the carboxylic groups, mostly with glutamic or aspartic residues of your RGD. Okay, so this is how your cells will attach with your collagen or gelatin. Next question. So which technique is non-optical microscopy? Non-optical means something which does not use visible light, or something which does not use lens to form an image. Okay, so A, dark field microscope, B, confocal microscope, C, bright field microscope, and D, atomic force microscope. What could be the answer for this? Anybody? Yeah, tell me. Which technique is a non-optical microscopy? So I told you, right? Non-optical means where you don't need visible light, like how normal microscopes work. You need a light. The light passes through the sample, and from the sample it goes to the lens, and from the lens it goes to the objective lens, the objective to then your camera, or wherever the image has to be made at the.
Yes.
Which one is not this type of
Option D.
microscope? Option? Someone?
Option D.
Yes,
D.
very good, very good. So, atomic force microscopy is not this type of microscopy. So, it is non-optical, which means it does not need visible light, or it does not need a lens for its function. Okay, so atomic force microscopes are based on a unique non-optical surface interrogation technique. This is built on the fundamentals of scanning probe microscopy, which utilizes a physical probe to measure the surface features of the sample with atomic resolution for lateral and height measurements. So, non optical interrogation technique means it will interrogate the surface of your sample. So, if you want to know the topography of the sample. It's called topography. So which means whether there is like where there is depth, where there is height, what is how large, if there is a height somewhere or a bump somewhere, how large is it spread on the surface, where there is narrower area, something like that. So how it is done? It is built on the fundamentals of scanning probe microscope. Scanning probe means you will have a tiny probe. It will be made of some material which is compatible to move your whatever incoming source is there for detection. So along with that, the probe also will move. So that time the probing is nothing but it will go and physically touch and it will detect like this. Okay, so it is a physical probe to measure the surface features of the samples at atomic resolution. You have to be very careful. It is atomic resolution. That means in Armstrongs or in nanometers, it will give you the resolution that good that sharp or that uh, tiny the probe and the feature of its measurement is. Okay. So this is the principle of it. I'll just show you a diagram. So here you can see this is the basic uh, requirements for it. So this is your quadrant diode, or your it's like a photo multiplier or something where it will collect the signal from your sample. Okay, and this is your laser. So this is your source of light or uh, energy, whatever it is, because here you don't use visible light to capture an image or something. So here you use laser light, because laser light, this has to be at atomic resolution, so the light has to be very sharp and very pointed, and it should not spread and diffract and deflect. And so you know how sharp laser lights are, so you use laser light here. And then you have a stage like this. This is a cantilever, and this is your stage for your sample. Okay, this is your stage for your sample, and this is your sample. So the image you excuse me, the image you get here, you can see, will look something like this only. So this is how it will look. So when your surface is straight, it will have this kind of a depth density or a depth measurement. And then when you have a bump on it, then you will have a measurement of this. It is color gradient, so that you can see the difference in the depths of it. Okay, so here you can see this one. So this is your probe they were talking about. So this is your physical probe, which is a sharp pointed tip. Okay, so but it is also flexible. So you should remember, when it has to probe and it has to move across it, it has to be flexible. If it's sharp and rigid, it will break, and then there is no point in moving it. So this is also movable. This cantilever is movable, and your stage sample is also movable. So you will get a full area scanning. Okay, so this probe is, as I said, it is flexible. So what happens? So it's like flexible like this. So when it goes and touches, like you can see my hand, it's like bumpy. So when it goes and touches, here it is straight, it is straight. Here it is bumpy. So it will go like this. It will bend here, right? So here it will be going straight. Here there is a bump, but it has to still move from it. So it will bend like this and move. So your signal which goes from here, so your bending of your probe will help bend your laser beam that is emitting out from it also. So then, based on the angle or extent of bending of the probe, your deflection of your laser light also will have a angle. So this angle and this will be caught by your diode or your collector. Okay, so sharp tip mounted on a cantilever scans the entire surface. A soft cantilever bends because of the tip sample forces. So when your tip and sample they hit each other, it will have some force during the movement. So based on this force, your cantilever, I told you this cantilever is also movable. So based on this force, your cantilever will also move like this. So if your tip is like this, your cantilever is here. So if your tip is bending like this, your cantilever also will move. So based on this movement only, there will be deflection in your or change in your uh, deflected laser. So the laser light deflected from your cantilever is captured on a sensor, that is your photodiode. It's a sensor, it collects the light. So your piezo actuators are used to move the tip or the sample in 3 dimensions, allowing scanning of the sample. So your piezo will be somewhere here, I think. The piezo are used to move the tip or the sample. So those are like nothing but levers or sorts. So it can either move your tip or it can move your stage. So when you move your stage also, it will have like a movement. It will move in x direction, y direction and z direction. So in 3 dimensional it will move. So this is your x, this is your y, this is your z when you're doing your microscopy. So it will go like this once, it will go like this, and it can go like this also. So that time you can see the depth in areas and you can scan the entire region. Okay, allowing the surface akin to a record player. So whatever this sensor is collecting, this will be later recorded in your instrument, and then it will calculate. It will have some mathematical models and formulas, and then it will calculate and it will give you uh, graphs, or it will give you these kind of topography images about the uh, surface features of your sample. So it can be operated in air on a buffer. So since it is a physical probe, you can either make it move directly in your air if it's a dry sample, or it can also move in your buffer if it's a live sample. Okay. So if anybody has any doubt, please ask me. I will repeat it again also if you want. Okay. So this is your atomic force microscopy. So this microscopy is very, very uh, highly important because the surface features you cannot know with your normal microscopes. You can just know that there is some bump or there is some elevation at certain area, but what is the measurement, what are the numbers, what is the length of it, you cannot know. So for that you have to go with atomic force microscopy. Okay, there is another one which is scanning tunnel microscopy, I think. So there also you can do the scanning, but there you cannot get the depth. The depth is not much, so you can only see like a scan a particular area. Okay, so yeah, atomic force has this depth analysis feature. Okay, next question. X-ray photoelectron spectroscopy is used to analyze what? A. Chemical constituents at the surface of the material. B. Topography of the surface. C. Chemical constituents in the bulk material. Or D. Topography of the bulk. What could be the answer for this?
A.
A, very good. So, X-ray photon electron spectroscopy is used for your chemical composition or whatever chemical elements are there at the surface of your material. So here you can see that it's only for the surface. It's not for your like huge or a bulk material. It cannot identify everything that is inside. So it's when you have a heterogeneous sample where at different layers you have different molecules, biomolecules, then whatever is on the surface, it will have a particular length wherein it can measure, because it's photoelectrons, so the electron have to penetrate into it and then eject out. So what is the depth of that electron penetration? It can measure only that much. So that is the reason why it cannot measure the entire sample in bulk. Okay, so your answer for this is A. Chemical constituents at the surface of the material is analyzed by X-ray photoelectron spectroscopy. So what it is an X-ray beam is directed at a solid material which ejects electron from the top layer. So when the X-ray beam hits on the top layer of material, like I said, the electrons are ejected out from the top layer. So how much the penetration power is there of your beam, then that only from that area your electrons can be ejected out. A spectrometer measures the kinetic energy of these electrons. Then it will have a spectrometer where it will measure the kinetic energy of these electrons. So when these electrons are ejecting out, it will have specific kinetic energy when they are moving out or getting ejected. So this kinetic energy is measured by your spectrophotometer, which allows the binding energy of your electrons to be calculated. And then using this kinetic energy, the instrument will calculate the binding energy of the electrons. The binding energy is related to the strength and nature of the chemical bonds. So the binding energy, for a chemical bond, you need to know what is the binding energy. Every chemical bond has a different binding energy, and based on the binding energy, it will have its stability. More the stability, more the binding energy, something like that. So based on this calculation of the binding energy, and every particular chemical will have a particular binding energy. So based on this binding energy, now you will have to skim down which bond might have been broken, because it has ejected out this much electrons, which resulted in this much of binding energy. So this binding energy equals to which molecule. So in that way, it will find out what is the chemicals that are present in that area from where the electrons are ejected. XPS, that is nothing but X-ray photoelectron spectroscopy, can measure the elemental composition. That is, what all chemicals or what all molecules, type of molecules are present in that, or the chemical state and the electronic state of the material. Electronic is like how positively charged or how negative, what is the ionic state of the material. And it can also be used to determine the relative abundance of element on the surface. Now you know that there is a particular element in this surface, because and because of the binding energy, because of the electrons that can be ejected out, and because of the capacity of that electrons to be ejected out, you can know that the same binding energy is coming out how many times by how many electrons. So based on this, you can also know the abundance. Not only you can not only say that there is like some carbon present in it, you can also say how much of carbon is present, but in that limited area where the X-ray can penetrate. Okay, so it can give a relative abundance of each element on the surface. So this measurement is also not like point measurement, it's like a relative abundance. Like if there is a composition, you have 10 elements, or the total number of elements or total number of molecules is some 100, and the different types of elements present are 10. So out of these 100, how many are type 1, how many are type 2, how what percentage is type 3. So it will measure a relative abundance like that. Okay. um, yeah, so this is how it works. So this is your sample. They're usually solid because they will focus the beam in a high vacuum uh, environment. So in vacuum, you know what happens to liquids. So mostly it's done for only solid samples. So when there is high vacuum in solid samples, they will focus X-ray beams with a power of 1.5 kilovolts. Okay, so this 1.5 kilovolts will go and hit the sample. So you see a particular area that it has hit. So that is your focus area, and then the depth which it goes, and then from this area, wherever the beam has hit, it will start ejecting out electrons, because it has been hit with a high volt X-ray beam. So it will now in turn throw out its outermost electrons. So now your electrons are out, and now see, every sample will have angle of that electron ejection. So that is your electron takeoff angle. And then the photo emitted electrons escape only from the very top surface that we understood. So 70 to 110 angstroms of the samples you can see. So this much is the depth in which you can use this. And then this ejected electrons will be taken up by this electron collection lens. Okay, so here you have an electron energy analyzer measuring the kinetic energy of the electrons. Now you have given it focused X-rays in a particular energy, that is your 1.5 kilovolt energy. Now based, so this is your standard, this is your constant for everywhere, all regions. But even though you're giving standard energy, standard amount of energy, every electron that is getting out or every electron that is ejecting out will have a particular binding energy based on the chemical that it is getting ejected out from, based on the chemical bond which has been broken or which has been there, from which it has come out. So based on that binding energy, it will have one force or it will have one kinetic energy with which it will eject out. So that kinetic energy will be measured by this analyzer by collection of the electrons. After finding out the kinetic energy, here only you can then convert it to a binding energy. Then the electron detector counts the number of electrons. So you have to know how many number of electrons are coming out, right? Because based on that you can know the abundance also. You can know, how many same types of electrons are coming out, how many different types of. So you have to have your electron detector which counts the electron as well. Then from here it will go into your this XPS signal graph from a silicon wafer. These are all internal things, not really required. So what you have to notice here is you should see how your result comes out here, how your output is. So your output comes out as your signals, like your peaks, like in your any structural measurement or structural characterizing instruments like your CD or your spectroscopy. This also uses a spectroscopy, right? So your output also will be in the same model. So you will get peaks like this, you will get elevations. So this is your amount of binding energy of this and this and this. So based on that peak, you will know which is which chemical or which is which element. Okay. So this is how your X-ray spectroscopy works. So if this was a little difficult to understand the basic principle behind it, I have a simple diagram here. So this is your sample. This entire thing is your sample. Okay. But it can only measure this blue color region. This is your pale blue, which it cannot measure. This is your sky blue color that it can measure. That too, this is because it is the depth in which the X-ray can penetrate. That will be in some microns depth only, not more than that. So micrometers. So within this micrometers, now your electrons can only come out of your outermost, even within that some few microns in which your X-ray can penetrate and hit. Your electrons can come only from the topmost layer. So that topmost layer is only your 70 to 110 angstroms depth. So you can imagine how superficial and how surface oriented information you can get from this. Okay. So because this is at atomic resolution, that information itself is huge. So when your X-ray rays come and hit the sample, it will penetrate, and then from this topmost dark blue layer, your electrons will get ejected. So the photoelectron characteristic of the sample surface. So that means, like how I said, the based on its sample, each electron will eject out at a different angle and it will have a different kinetic energy. Based on that, you can back calculate the binding energy. Based on the binding energy, again you can go and detect or you can filter out which element it is from. Okay, so this is your basic of your X-ray photoelectron spectroscopy. Okay. um, Next question. Contact angle on the surface is indicative of dash and is measured using dash. Contact angle, I think we did in the last session. Okay, so somebody
E,
should answer. Yeah, someone said something.
E,
Now
option C, ma'am.
C. Yes, yes, very good. So C is your hydrophilicity or hydrophobicity. You can, either way is correct. So it can either be hydrophobic or hydrophilic. So generally we say hydrophobicity, or then you can also say hydrophilicity. So your hydrophilicity is the property that is measured using contact angle on the surface, and the instrument that measure is called as goniometer. Okay, so what is a contact angle? uh, See, you can see in the image when a liquid falls on a plastic surface or any surface, solid surface, every liquid droplet or a drop will have a particular shape that it will attain. So you can see based on the shape of it, it will have a contact angle. Contact angle is nothing but the angle between your surface of your liquid, horizontal surface of your liquid, and your surface. Okay, so here you can see if your contact angle is less than 90 degrees, then your surface is hydrophilic. If your contact angle is greater than 90 degrees, then it is a hydrophobic surface. If your contact angle is greater than 150 degrees, then it is a super hydrophobic surface. So we discussed this in the last session. So here, why does this hydrophobicity or hydrophilicity come? Because based on this hydrophobic and hydrophilic nature only, your liquid can go and attach to your surface. So if your surface is more hydrophobic in nature, it will repel your liquid, so your liquid can't go and sit nicely on it. It's also called as wettability of the surface. Okay, so wettability, based on this, how your water droplet or your liquid droplet reacts or how it sits on your surface is very important to know the surface hydrophobicity or hydrophilicity. Why this is very important? Because based on this, you can have materials of different hydrophobicity. Like you might have seen some videos, like I said in the previous, you have might have seen some advertisements where they say that this is water resistant material, or this shirt can, even if you throw like a cup of water on it, all the water flows down, it doesn't get absorbed into the material. So that is because they are like super hydrophobic, so they have a contact angle much higher than 90 degrees. It's more than 150 degrees. So that is the reason the water cannot even go and sit on it first properly to get absorbed. So that kind of property will help the material in many different ways. So if the plastic surface has high surface energy, then the liquid will spread. So if there is high surface energy, means it is more hydrophilic in nature. So that time it will have more high surface energy. So that surface energy will allow your liquid to settle on it and nicely spread on it. But if your surface has a very low surface energy, then your liquid cannot, even though it has fallen on the surface and it is straight, it is flat, your droplet will not be able to spread out because there is very less surface energy that is not allowing your droplet to, spread on the surface. So it will bead up. Bead up means it will, when your falls, it will acquire that spherical nature. So that spherical nature is beading up. So when it beads up or it looks very spherical in nature, that means the surface is very hydrophobic. Okay. So this angle, as I told you, the angle between the outermost surface of the drop and the surface of your surface of your base or your stage or your solid on whatever it is, that angle, this angle, and this angle, these angles are called as contact angles. So if the droplet spreads, wetting a large area of the surface, then the contact angle is less than 90 degrees. So if it spreads, obviously it will become flat like this, right? So when it becomes flat like this, you can see the contact angle is very steep. It's very narrow. It's it's less than 90. This is your 90-degree line, perpendicular. So when you draw like this angle is coming less than 90. So that time it will spread when it becomes flat, so angle will become narrow. And then your surface is considered to be hydrophilic or water loving. If the water drop forms an angle greater than 90 degrees like here, then the surface is highly hydrophobic. the water tends to bead up on the hydrophobic surface and it will not go flat. Okay. So this is your goniometer. So this is your surface where you want to check the hydrophilicity or hydrophobicity based on your contact angle. So what this instrument will do is it will measure the contact angle between the drop and the surface. So based on that, you should know how hydrophobic or hydrophilic the surface is. So here you can see it has a camera, it has a surface, and this is your light source. And then you'll put your water drop, your standard whatever liquid you use for it. Then your camera will capture the image and then it will look something like this. So you can see there is sample A, there is sample B. Okay, these are two different types of samples. So here you can see your sample is like this and your second sample is like this. So how do you measure your contact angle? You cannot directly just go and draw like this using a scale or something, because these are very tiny measurements. So you have to have this microscopic measurement. You can see this white boxes or dots they have put. They will measure it between this and this, and then the resolution has to be very high to measure this accurately. So what they do is they have developed mathematical models to measure this. So either they will use polynomial fitting method or ellipse fitting method based on how your sample is sitting on it. Now here you can see your sample is sitting nicely flat like this. Here you can see your sample is sitting somewhat in a curvature kind of thing. It's not even properly a full bead on top, but it's still going at the bottom. And then you can see some curvature of your surface due to some surface energy interaction. So because of this, here, because your angle is not very steep and sharp, you will use this ellipse fitting model. Here you will use your polynomial fitting method. If you want to know more information about these models or methods, or how this camera works and how this liquids are contact angles are measured, you can go and visit this paper from where I have taken this information. It will be useful. Okay, so now based on all this information, you have guessed this instrument, you have this hydrophilicity, the contact angle. Can you tell me, between A and B here, which one is more hydrophilic according to you, if you have to just guess it? Anybody? Between A and B in this image, which drop is telling you that your surface is more hydrophilic? Come on,
Might
might
be a,
be sorry.
might be a.
A, yeah, very good. Why? Because your droplet is very flat on this. So your contact angle is less than 90 degrees. You can see. If I draw a straight line here, your contact angle will come like this. It's almost 45 degrees. It's less than even 45 degrees. So this surface is highly hydrophilic in nature. Okay, very good. Someone is listening. Okay. So shall we go to the next question, or does anybody have any doubts in this? Anybody? No? Okay. So next question is, hmm, I'm so sorry. Okay, I think you have seen the answer. Anyways, uh, the question is, dash binds to the mannose residues of various glycoproteins and activates lymphocytes. A. Glycoprotein. B. Concanvalin A. C. Pectin. And D. Nucleosides. So, what is the answer for this? Come on, tell me. The answer had been already revealed. You can tell the answer. Anyone? Yes. Oh, you guys are answering in the chat box. I told you I won't be looking at the chat box. Please open your mic and speak up. This is a live session. It's supposed to be interactive. I'm so sorry, guys. I have not seen all these answers. Now I don't know which option is for which question. Okay. Anyways, so what is the answer for this question now? What is this question? What is the answer for this? Dash binds to the mannose residues of various glycoproteins and activates the lymphocytes. Anybody? Come on, speak up. I'm not going to move until you guys speak up.
Option B.
Option B. B for ball.
Option B. Yes, ma'am. Balls.
Okay.
Concanavalin.
Yes, very good. So the answer is option B, concavalin. So what is concavalin? So it is a lectin. Okay. A lectin is nothing but a protein. Okay. Lectin is a protein. Okay. Lectin is also, I think, the first protein of whose structure was known, something like that. Okay. So
Leptin might be carbohydrate.
sorry, your voice is breaking.
Leptin might be carbohydrate.
Oh, lectin is a carbohydrate. Oh, I'm so sorry. Okay. Okay. So you can check if lectin is a protein or carbohydrate. I'm not really sure. So this can be like your extra question. So concavalin is a lectin that is used in cell culture or to stimulate the T cells and study cell proliferation and differentiation. So con A, that is concavalin A, is derived from a plant called as Canavalia ensiformis. Okay, this is the scientific name of the plant. So what does this do actually? So what it does is it is an alternative for your antigen presenting cells. So antigen presenting cells, right? What does it do? It will help in exposing the antigenic part of your antigen or your foreign particle or your pathogen or any microbe which is not supposed to be in your body, anything that is foreign to your body. It will have an antigenic region. So the antigen presenting cell will process this region and it will present it to your uh, immune cells like your T lymphocytes or your B lymphocytes or your other natural killer cells like that. So how does it do? It does it with the help of the MHC proteins. Okay, so your concavalin is working somewhat like your antigen presenting cell and helping your T lymphocyte to identify these surface glycoproteins. Okay, so here is your endothelial cell. So this your endothelial cell has this glycoprotein. So it depends on what type of cell it is. It may be some cancerous cell or some other cell which is not supposed to be there. Something like that, some injured cells where your immune system has to act now. So if you inject concavalin to your animal model, then this will go and attach to this glycoprotein surface, and then it will signal your T cell to come and identify this as foreign and show some immune response to it. So if it's your T cell, it will show your T cell response, or if it's your B cell, it will produce some antibodies. Okay, if it's your NK cells, it will kill the cells immediately. Your cancer cells. So here you can see some signaling molecules that are being released here, like TNF alpha or IFN gamma. All these are pro-inflammatory secretogens. Okay, so your other cells like your Kupffer cells, these are also your immune cells, tissue immune cells. So these cells will usually do this presenting of this foreign cells or your injured cells or something. So in place of this, your concanvaline will also do the similar type of cell. So then later what happens, it will either go to apoptosis or it will have some pro-inflammatory effects on the cells. So majorly why we use this is more specific to your T lymphocyte. So when you're doing some experiments with your T lymphocytes or some differentiation or you're growing your cells, so it will help in your proliferation of these cells. It is also a mitogen. Okay, mitogen is something that induces your cell cycle. So it will help in proliferation of your T lymphocytes. The applications, few of the applications of your con A are written here. One is your T cell simulation. Con A is used as an alternative to antigen presenting cells to stimulate T cells. It binds to glycoproteins on the cell surface, which causes the T cells to proliferate. It is an antigen independent mitogen. Okay, so then it also is used for cell proliferation and differentiation, where it can improve the proliferation and differentiation of various animal cells such as dental pulp stem cells. Okay, and hormone receptor studies. Con A is used in hormone receptor studies, and it is also used in characterization of cells, whether they are normal or they are malignant cells. Malignant are your tumor cells. Okay. um, there's one more slide. Yeah, so you can see here, um, so this is how your concavalin is used in the research. So in vivo conditions, you can take your model, which might have, in which you might be doing some tumor studies or tumor analysis. And then if you inject it with con A, where it has tumor, there will be activation and expansion of CD8 positive uh, cytotoxic T lymphocytes. So these will help in killing these tumor cells. Then it will also activate NK cells, natural killer cells. These will also act on the tumor cells. So then you can see that your mammal which had this tumor or your animal model will have a reduction in the tumor nodules. Okay, then it can also be done in vitro. Like I said, if you're culturing the T lymphocytes, or if you want to do some experiments with the T lymphocytes, then you can give a con A treatment. Cancer specific, they have used cancer specific T lymphocytes here. You can see CD8 positive cells are there. So then the con A will help in proliferation of these cells. Then when these cells proliferate, they'll get activated, and then you can use this again on some cancer cells to see the cancer specific lysis or some cell specific lysis, that which type of molecules it is attaching or which type of cancer cells it is more effective on, such kind of studies. And then in chemotherapy ex vivo, so con A coated cancer cells are given in the injection to cornicoated injection that cornicoated cancer cells. Yeah, so they are injected here, and then injection viable cancer cells are taken, and then ex vivo cancer specific cell lysis is there. So here what they are trying to do is, so here you saw that I told you, like, few cells might be more specific to these T lymphocytes that are activated by cornea, and then few might not be. So here also you see they have written reduced tumor nodules. They have not said that complete tumor is gone or completely tumor is eradicated. So here what you can do, you can, because this cornea has specificity to certain cells, so that means there are other cells which are still viable cancer cells which are still living. So then what they will do, first they will kill whatever is susceptible to cornea, and then viable cancer cells will be taken, and then specific lysis will be tested or analyzed, or if they have some other drug that they have developed which is more efficient than this, they will do the comparison studies, etc. Okay, so that is your con A. I'm so sorry, I don't know if it's a protein or a carbohydrate, please check. Okay, so your next question. Yeah. So, so your next question is, Retina contains rod and cone cells. Rods detect dash and cones detect dash of the light. Anyone? Please answer.
Option D. Option D.
Option D. One second. Yeah, there is some internet issue. So you guys said option D. So that means retina detects rods detect intensity and cones detect light, right? Yeah. So this is a typo. It's D. So it is intensity and color. Okay. So write it. Okay. Mm, Yeah. So the human retina has two types of photoreceptors. So your rods and your cones are called as photoreceptors because they uh, collect light. Light is your photons. So they are photoreceptors and they gather light, and they are named as rods and cones. While rods are responsible for vision at low light levels, second, cones are responsible for vision at higher light levels. The light levels where both are functional are is known as mesopic. Okay, so your rods which detect your intensity are responsible for your low light visions, and your cones are responsible when you have high light or more light. So that is why when you have more light or in daytime during bright light, you can see lot of colors, you can differentiate between colors. But when the light is very low, you cannot really differentiate between colors, and you will only see shades of gray, like you, that is because of your intensity. So whenever you see something in grayscale, if the intensities are different, then you can see the difference. Otherwise, you can't make out. Okay. So what are the main differences between rods and cones? These are responsible for vision in dim light. Cones are responsible for color light and works best in bright. Just now we discussed. Then the pigment in rods is rhodopsin, which is formed from a protein called as scotopsin, and retinal. Okay, retinal is an aldehyde form of vitamin A. Then your cone cells, the pigment present in the cones is photopsin, and it is formed from opsin protein and retinal. So this is rhodopsin, rod cells, photopsin, cone cells. Okay. Um, there are about 120 million rod cells, and there may be some 6 to 7 million cone cells. Rods are predominant in the extrafovea region, and cones are concentrated in the fovea region. So this fovea region is in your eye, where your cone cells are present, and the region that is outside your fovea region is your extrafovea region, where the rod cells are present. So this is your eyeball, and this is your retina, and uh, this cover lining you see, this lining is where your rods and cones are present. So this tiny square they have depicted here, and then they have enlarged it, tiny section. So here you can see these are your rod cells, and these are your cone cells. And then the cone cells are of three types. They are which collect your blue, green, and red, and they are called as short, medium, long. Okay, so this is your rods and cones of your eyes, of your retina, which helps you in detecting and analyzing your different intensities of light and also different colors. Okay. Next question. Rods and cones are dash. A. Electrically active. B. Sound sensors. C. Pressure sensors. D. Electrically inactive.
Electrically active.
Electrically active. Very good. So the answer is A. Why are they electrically active? Because they convert the light into electrical signals. Okay, so rod and cone cells are light sensitive cells in the retina that convert the light into electrical signals, and these signals are then transmitted to the brain via your optic nerve. So just beside your or next to your rods and cones, that fovea region and extrafovea region is your optic nerve. So the signals from your rods and cones is then sent to your optic nerve, which will then send signals to your brain, and then your brain will process the electrical signals and form a image or project an image in your brain. And when the light hits the retina, how does this happen? When the light hits the retina, the rods and cone react by undergoing a series of chemical reactions. So this opsin, photopsin, rhodopsin, they have this all this chemical reactions going on, and then the brain then interprets these signals from the rods and cones to create an image. This chemical reaction is then converted to electrical signals. Electrical signals reach your brain will then create an image. So this is your light. This is how your light hits your eye, and then it from here it will reach a focus point on your uh, rods and cones, where your rods and your cones will collect the light and intensity and color, and then you can see the optic nerve here. So this information will be processed by your rods and cones, and then it will send a signal to your optic nerve, and then the optic nerve will send a signal to your brain. Okay, so this image, light micrograph image I wanted to show you. So this is a cross section of your photoreceptors or the region over there. So this is how they look in real life. So you can see how tightly packed they are, and then you can see the size difference of these cells. So you can see here, right, something, you can see this. So that is only this. So these are your S cones, those are your short cones, and then these are your L cones and cones, that is your long and medium cones, and then these are your rod cells. Okay, so this is taken from a monkey's retina, that is your apes. So this is from there. Humans is also very similar and same as these, and these are unstained cells. So you can see that your cones have pigment in them, so they are appearing darker in color. Okay, so you can know how this your light is getting converted to your image if you can think of it like a camera. So you in your camera you will have sensors, right? You'll have sensors which will collect the light, and then that collected light will be having it will be processed, and it will be having some codes within the chip or within the motherboard. So this coded signal will again go to your microchip in your camera. So your then chip will process that based on their language, and then it will then project an image in your camera. In the same way, your eye also will capture this light. It will transform this into some coded electrical signals, and then it will send it to your optic nerve. So your retina acts as your sensor, and then your signals are sent via your optic nerve, and then your brain acts as your chip, which will process the code and then form a final image. Okay, um, this is about your rods and cones. Last question. Dash is used in the cell culture to dissociate the cells and tissues and isolate primary cells. A. EDTA, B. Laminin, C. Collagen, and D. Collagenase. What is the answer? Anybody? What is the answer for this? I think we didn't discuss this, but you might be knowing one of them as a dissociating agent that is used in cell culture. Anybody? Okay, what is going on? Is anyone there? Why are you guys not answering? Last question, come on. Which one is a dissociating agent? EDTA, collagen, collagenase, or laminin? Can someone answer?
EDTA.
EDT.
EDTA. Okay. Anyone else with any other answer? No? Okay. So it's not EDTA. I kind of knew that this might confuse you, this EDTA option. So EDTA, I think you are telling on the basis of trypsin EDTA, which we discussed before. So if it is because of that, then I would like to remind you that EDTA helps in activation of the trypsin, but EDTA will not cleave. Okay, trypsin is the one that cleaves the cells from their attachment. So EDTA is a calcium chelator. So it will help in, chelator means it will it will attach to all the calcium. Okay, so it will make the calcium unavailable to other things. So when this happens, then your trypsin gets activated. So EDTA will help in your trypsin activation, but trypsin is the dissociating agent. So it's not it's not EDTA. And then laminin is your ECM protein. Collagen is also your ECM protein. What is collagenase? Collagenase is an enzyme. Anything that has A's in it, you should consider, you should you should expect, or you should guess that it's an enzyme. Okay, protease. It's a protease. Okay, so collagenase will help in cleaving the collagen. Collagen is the one that helps in binding of your cells to the ECM or your basement membrane or your plates, right? So the collagenase you can use to cleave this collagen. So when it cleaves it, your attachment will also be cleaved. Okay, so we can use both trypsin and collagen. Few people prefer trypsin, few people prefer collagenase. Okay, so collagenases are enzymes that break the peptide bonds in collagen. Different types of collagenase are available, including type 1, type 2, type 4, type 5. These are the types of collagenases that are used. Okay, so different collagenases are used for different tissues. So to isolate cells from fat, that is your adipose tissue, your lungs, your epithelial cells, and your adrenal tissue, mostly we use type 1. To separate tissues from liver, thyroid, bone, heart, or salivary, mostly glandular cells or bone cells or heart cells, you use type 2. And then type 4 is used for your islets from your pancreas, and your type 5 is also used for your pancreatic islet tissue. Okay, so the storage for your collagenase is 2 to 8 degrees if it is powdered form, and you have to protect it from sunlight. So you have to give it in some amber tube or some opaque container. So when it is reconstituted, you can store it in -20. Okay, I so since we are talking about trypsin and collagenase, we as I was saying, you can use them all as alternatives. You can see here there was one study that they had done to see at for a particular skin tissue like keratin. They have keratinocytes they have taken and the tissue, and they have isolated it using dispase 2 or trypsin EDTA, and then they have isolated using collagenase. So they wanted to see if there is any difference after isolation and in the viability of the cells, and then after that when they grow it in vitro, then if there is any difference. Okay, so this is the tissue section from where they have taken it. These two regions of the cells, these are plated on these four sections, they are plated on the cell culture plates. So this tissue region was trypsinized, and this tissue region was dissociated using collagenase. So can you see any difference in the cells here that are being grown on the surface? So what they have seen in their particular cell type, that is for their skin cells, is when it was dissociated with collagen, because it is a biomolecule, it is a biological enzyme. Trypsin is chemical, so that time the cells were more, once again, when they were seeded in the cell culture dish, they were more compactly arranged. You can see the green region is your cytoplasmic region, the red is the nucleus of the cell. So you can see here that it is more compactly arranged when they were collagenized. But when they were trypsinized, they are still little, they are attached to each other even in the dish, but they are still little far apart from each other, and they are spreading more than usual. So here also you can see their arrangement, how it is. So based on the cell type, you can choose whether you want to use trypsin or trypsin EDTA and then collagenase. Again, there is also trypsin alone without EDTA which can be used. So that is not recommended, because trypsin alone is a little toxic to the cells. It might, autolyse the cells, so it will lyse the required proteins along with the detachment of the cells. So usually you go with trypsin EDTA, and that too you go with very low percentage of trypsin, that is 0.05% to 0.25. Maximum is 0.25 we go. Above that, if you go, it is a little lytic to the cells as well. So we generally go with less than 0.25. Mostly 0.05 works very well with most of the cells. And you should also remember that after adding it to the cells, you have to wait for a couple of minutes, 2 to 4 minutes, but not more. If you wait for more time, then autolysis will occur. So that is why after you are done with your 2 to 4 minutes incubation, you take it out, and then immediately you neutralize with your serum media, that is your complete media. So the serum components of your media will help in deactivating your trypsin. So that is how you detach your cells, and again you seed it and see that they are in a well maintained healthy condition after the process of isolation from your primary site of tissue. Okay, because during isolation, if there is any disruption to the cell, and then they have been injured, then when you see them, you start growing injured cells, then they become senescent, and then they start dying. So isolation process is also very important. Okay, so I think that's it for today. Any questions, anyone? If anybody has any questions, you can go ahead and ask me. Or if you want me to, revisit any of the questions you didn't understand anything. Retina, concave, A.
Ma'am,
Till where I know, lectin is a protein only. So lectins are proteins. Is
ma'am,
one of
lectin, lectin, ma'am,
yeah, yeah. Tell me.
lectin. Lectins are actually proteins that binds to certain carbohydrates.
Yeah, that is what I was saying, but then someone said it is a carbohydrate. So that's what lectin is a protein that binds to carbohydrates. So it's generally called as carbohydrate binding protein. So maybe because of that you got confused. So
Yeah.
yeah, it's fine, no problem. So whenever there is a confusion like that, we can always go and recheck. So why I am saying this, because lectin is, I think, one of the first proteins whose structure was solved. So it's a protein. So like that, if you have any other doubts, you can ask me. I will, if not now, I can clarify it in the next session as well. I can also do some research if I don't know about it, or you can put it up on the portal. We can, look it up and give you the correct answer. So yeah, so lectin is a protein, carbohydrate binding protein. Where is it? So your concave is a type of lectin. Okay. So other than this, anything else? Contact angle, XPS,
Oh man,
AFM. That's it. Okay. So I think we are done for this session. Thank you for attending the session, everybody. We can close the session.
thank
Thank
you.
you, ma'am.
Thank you, ma'am.